On that note, try to develop a mind of expectancy. Get up early in the morning, 1 hour every day will give you an average of 6 hours every week, give you an extra by 52 weeks or 50 something weeks of the year. And that is an amazing feat. For us in IMG, this is IMG Research Academy, where we talk about clinical research, where we also try to demystify clinical research with the aim or the objective to improve patient outcome. Every now and then we have these clinical talks or research talks where we pick a topic in clinical research, we get a guest speaker that will talk to us and try to demystify it, try to reduce it to the most simple term that we can imagine. Understanding clinical research is a big thing. And then it takes people that have done it over and over again to be able to talk about it in a very simple way that a little baby can understand. Because if the little baby doctor cannot understand it, then how then can you conduct this research to improve the lives of your patient? My name is Edward Tokobi, and joining us today all the way from the other side of the world is Professor Khalil. I'll allow him to talk about his profile. He's been an experienced clinical research statistician and also statistician in other spheres of life. So he understands what we're going to talk about today. In clinical research, there are two types of data. There are two types of study depending on the availability of data, the primary study and the secondary study. The primary study is where researchers go and get data. The secondary study is where we use already existing data to refine and form ideas. One of those there are different types of secondary study, the literature review, the systematic literature review, and others. One of them is a meta-analysis, which fortunately is almost a zenith of all clinical research, where we collate and aggregate data that is from different homogeneous studies to see if it makes sense to the common man, if what we found in this single study, is it translatable, or is it juxtaposable, or does it have a size effect? What is the size effect to it? I wouldn't talk much because we have a professor in our midst, and I would like him to shine today, as he has always done. He's been my friend for a very long time, and today I will be introducing you to him. He will be going into details of meta-analysis and how to conduct a meta-analysis. He may not be able to cover everything because we want to reduce this talk to just 1 hour where it will be very focused. So he will be giving us high-level details, and then your questions and interactions. We encourage open questions. You can stop him anytime in his presentation and ask your questions. Professor Khalil, you can unmute yourself and tell us about yourself and then jump right straight into the conversation. You can unmute yourself, sir. You can unmute yourself, sir. Your camera, your microphone is still in the mute. Thank you.
Thank you very much, Professor Edward Tokobi.
Well, I'm not a professor yet, but that's like a prophecy. So I claim it. Someday we're going to get there.
Yes, yes. Okay. So I welcome all the participants from all corners of the world, and I am very pleased to interact with the medical doctors today in talk about A to Z meta-analysis. And I divide the talk in two portions. One, we will have a presentation on the meta-analysis, and after that I will share how I recently conducted meta-analysis. All the steps we will see practically, and at the end I welcome you for your questions. So
And I believe you want to share your slides. Let me know if you're having any technical difficulties in sharing your slides. As I have them, I could also share them from my end, but you could just open it on your laptop, click on the share button, share your screen, and then your slides will pop up.
Okay. Are you seeing the presentation?
Yes, we could see you. We could see your presentation.
Oh, that is nice. So let's start to talk about the meta-analysis. I am Professor Khalil Ahmed from Pakistan, Islamabad, and I have been working as a data analyst since from 2007, and this is actually a secondary data analysis. The meta-analysis is actually the analysis of analyses. So analysis of analysis, this idea was given by a great statistician, Sir Carl Pearson. And after that, some other researchers work on, and uh, where uh, Mr. John Hunter Frank, he has given so many statistics, and
And I
have
think your
to.
view is perfectly fine like this because we're seeing it perfectly.
Okay. Yes. Now it's better for me also. So this is the study material, and we can study a very comprehensive and about meta-analysis, how to conduct a meta-analysis, and it's and also the advanced meta-analysis. So the introduction to meta-analysis by Borenstein, Mitchell Borenstein, is very precise and comprehensive, uh, which has multiple chapters, but not we just like to the point. And after that, you can go for methods of meta-analysis by John Hunters. And when you will finish these two books, then you can go for the advanced meta-analysis, which is a structural equation modeling approach. So let's talk about what is meta-analysis. It's very precisely speaking analysis of analysis, because you uh, collect the data from already published papers, from uh, even unpublished paper, and from newspaper, any source which is uh, homogeneous and which is very related to your topic and to your problem. An objective and quantitative methodology for synthesizing previously studies and research on a particular topic into an overall finding is that meta-analysis allow us to collect, code, compare, combine the results from different studies and interpret using the statistical method, facilitating statistically guided decisions about the strength of the observed facts and the reliability of the results across a range of studies. Because we when we have uh, already published research, even unpublished research, significant results, and even non-significant results, the prime objective of any meta-analysis is to see the overall effect. You know that some research will, uh, if you collect one or two papers, it might be possible they are, uh, one is saying something is better and other is saying something is not better. Something has this adverse effects, and some is saying this is very safe. So meta-analysis will enable you to calculate the overall effect in totality. Is it better or not better, recommendable or not recommendable? So this is the reason, and it is also inevitable to average out the published research. So to provide a summary effect of all the previous studies, we have the tool available in the meta-analysis. So uh, it is very easy to find out the overall conclusion of the various studies. And previously, we go through the literature, we describe in a descriptive way, and now we are welcoming and we are appreciating the persons who did the systematic literature review. And the systematic literature review is the first step for the meta analysis. Efficient methods for claiming gaps in model and theory, tools for the theory development, and these are the reasons for which we go for the meta-analysis. Theory of planned behavior is given by, this is an example uh, for why we go for the meta-analysis. So uh, uh, next, we uh, have different uh, sources how we can conduct the meta-analysis. There are different softwares. Some are the open source software and some are the paid softwares. And I always recommend to go for open source softwares because they are updating time to time and uh, available in with full options. So uh, this is uh, some papers that may be very useful to study the role of meta-analysis, the theory of meta-analysis. And now when we go for the meta-analysis, the first topic is the first step, very best first step is to select the topic for which you want to search out the literature. And after specifying the topic, you will list out the important parameters or variable. Then you will go for uh, a systematic literature review. And while you are conducting the systematic literature review, meanwhile you will extract the numerical data in Excel file, in other spreadsheet files, to conduct the meta-analysis. You will also manage and the data extraction as well, with respect side by side with the systematic literature review. So systematic literature review, as I have already told you, that it's the first step to conduct the meta-analysis. So we have to develop some search terms and criteria selection to because when you go to some different databases, you will have too many number uh, on one topic. The research studies too many, and you narrow down according to your inclusion and exclusion criteria, which you will have to already decide which papers you will include in your meta-analysis and systematic literature review, and which paper you will exclude. So so this is the planning stage. And after when you have decide the topic, you have list out the variables or parameters which will project your study, on which you want to uh, calculate the overall effect and overall summary. And you have list out the inclusion criteria and exclusion criteria. And then uh, it is not always recommended as a single person to conduct a meta-analysis. It will be better if we have a team to conduct the meta-analysis. If someone did this review and other one will again cross check, which will be better, and it will increase the precision of the meta-analysis as well as the systematic literature review. So after conducting, while you are doing the systematic literature review, you will manage your data extraction process. So after that, conducting stage, and the last is the reporting stage. Reporting, you will when you will have extracted all the relevant data. Some paper will provide you and some paper will not provide you on some parameters. And after that, you will go for some softwares to conduct the numerical analysis, and you will write the descriptive report. So systematic literature review, you are already aware about that. And it's uh, literature review actually, but in a systematic way. So uh, uh, systematic literature review now can be categorized as hypothesis development, bibliometric analysis. This is not. This is also some journal appreciate if you also add the bibliometric analysis. And it is now we can do through different softwares, and it tells about how much research is published, where it is published, and so on. So our topic is in this talk is meta-analysis, and uh, and this is the content analysis, which is just like a thematic analysis. It's also uh, uh, there are different tools available to conduct the content analysis. It is also, it's we are now very technologically updated, and we have too many tools to conduct the content analysis. So now, the primary objective of this talk is we want to learn how to conduct a meta-analysis. So I have already told systematic first step is a systematic literature review. Second, compile the results of these studies in a format you can manage in Excel and other spreadsheet software. The numerical data on different parameters are variables which you have already listed out to find out the overall effect. Converting all these statistics to the effect size, and the fourth step is meta-analysis using the software. And after when you will uh, have done the meta-analysis using some software, you will get some numerical results like forest plots and funnel plots and model summary and some statistics about heterogeneity, some statistics about publication bias. So the last step is to write the descriptive interpretation and reporting of the results. So this is a sample study, and we have a sample format, and it's basically there are 5 studies of different 5 authors with the years, and this is the sample size, and this is the correlation coefficient. So you have seen this study told you the correlation is 0.5, and in this study it's 0.6, and in this study it's 0.4, and 2 studies are reporting the negative correlation. But in meta-analysis, we are always inclined to find out the overall effect. What is the overall effect? And this is the and how we will conduct. We will see.
So, Professor, remember where we said that
similarly.
Can you go back to the last slide? You know, how we said we're going to break it down in steps. So for many people that are, no, the next slide, next. Yeah, for many people that are very new in clinical research, they may not understand what R is and how those numbers were gotten. Do you want to shed more light on what R is, and that these numbers are actually found in the individual studies that you are trying to get, because there are many of us in different stages of research? Just maybe 1 or 2 seconds.
Yes, it's just an arbitrary example. It's a hypothetical example. Right. The correlation. Basically, correlation is the tells you about the association between 2 quantitative parameters. Either these parameters are associated or related, and if they are related and how much they are related, their strength of relationship. So the correlation coefficient tells about the strength of relationship between 2 parameters. It should be quantitative parameters. Like there is a relationship between height and weight, and we because these both are the quantitative variables, and we will find out the relationship through the correlation coefficient, and it lies between -1 and 1. So it's if it is 0.5, it means that there is a positive relationship, and it's a moderate have a moderate strength 0.5. If it is close to 1, we will say there is a very strong relationship between these two parameters. And if it is in a negative sign, it means that if one is increasing, other will decrease. There is a negative association or relationship. And if it is close to 0, and it means that the relationship is very uh, weak, very. So if it goes to close to -1, it means that there is association, which is very strong association in a negative direction. It means if one will increase, the variable will decrease.
Thank you so much.
So when we go through the paper of ABC, who wrote in 1989, there we find they have took 40 patients, and they analyze the correlation of some two variables, and they found the correlation or strength of association was 0.5. And another person who conduct the similar research in 1990, he took 90 patients, and the correlation was 0.16, and so on. But in meta-analysis, we want to find out what is the overall effect. Even some are reporting very low and in a negative correlation, and some are reporting positive and in a moderate relationship. And in meta-analysis, because it's a literature review, after that we want to study what is the overall effect, and it will uh, uh, put the researchers and push the researchers to search out to validate the new or to conduct the new research to find out what uh, the relationship is in the current scenario. So systematic literature review along with the meta-analysis is uh, uh, essential, uh, and it is recommendable to before to conduct the new research, to start the new clinical trials, and it will help you to develop your hypothesis for the very new research.
Thank you so much. We could
And
move to the next
sometimes,
slide. Yeah.
yeah, that was table. It means when you are uh, doing the systematic literature review, and you have to uh, manage the numerical data, the side by side data. This is the data extraction step. So if they have the mean and standard deviation in different, and we want to see what is the uh, overall effect between, it may be a single mean in different research study, and we will see the overall mean. And if they have two groups, like control group and treatment group, and they have the mean x1 and x2, these are the mean of you may say x1 is the mean of control group, and x2 is the mean of treatment group. So this is this SD, this is the standard deviation of first group or control group, and this is the standard deviation of the treatment group. And N1 is the sample size in control group, and N2 is the sample size in the treatment group. So you will, when you will go through the papers, you will also put the numeric in this table. After that, we will analyze the overall difference between treatment group and between treatment and control group. Either there is a significant effect of the treatment or not overall. There are variety of softwares available, and these are the Meta EC, Meta Excel, and Meta Essential, and these are the open source. And R Studio have the two packages, Meta and Meta 4 and Jamovie. And you can also use some other like Stata and uh, uh, there are so many softwares to conduct the meta-analysis, and comprehensive meta-analysis is basically paid. So this is the meta essential, and it is basically they have developed some sheets in Excel. This is very fantastic. You just put the numerical data into the Excel sheets, and you get your results. Uh, and they have did a very wonderful work, and I will share the link of Meta Essentia. They have write their user manual, how you will use this, and also how you will report the results. They have put the interpretations as well. It's I think I recommend the new persons who are going to uh, learn about the meta analysis. They should go through this website to go for their sheets of some different kinds of meta analysis. They have did and it's very precise. It's not in detail, and you will have a very brief report literature or about the meta analysis and how to report the results of the meta analysis. It will be very helpful. So if you go through the books which I have already mentioned in my very first slides, that is very perfect. And if you want to want if you want the summary of these books, and this is actually the summary of they have definitely extracted from those books. And if you go through the site, through the material available, and it will be very uh, uh, highly recommendable, and it will be very beneficial for all the learners. So this is also about the meta-analysis, and you will I it would be great if I put the link of this website.
So, Professor, now that we've gone through the phase of extracting the data, arranging them in an
Okay.
Excel sheet? What is in identifying the software? What is the next step?
Okay. So this is was the about the meta-analysis, meta-essential, how to interpret the results. You will find all these things, and you will download, and it will be a folder. There will be different sheets, and they will allow you just put the data on the sheets and get the results. And here are different tabs for different, like forest plot and so on. So the next after that, because you know that we have a variety of statistics while we are going through the papers, and
Yes, so
what
at this stage, I would like to inform all our members and those that are joining us, maybe that may also watch this video, that we're going to have a dedicated class to talk about the concepts and the terms in statistics, in medical statistics. And that class will cover what p-value means, what confidence interval, what forest plot means, what central tendency is. We're going to go through each and every one of them. And we look forward to that class.
okay.
Yes.
Okay, that is fantastic.
Yes, so that because if we go over them on this, we're going to spend so much time. So we have a class, someone has volunteered to take us through these different steps. And I believe Professor Kale will also be available to, weigh more of his thoughts when we are having that conversation.
Yes. So in this talk, I would like to conduct the meta-analysis in our this live session. It will be perfect as compared to show you this is the data and this is the forest plot. And
So, in the spirit of making it very simple, I think what these two data you are trying to do, but correct me if I'm wrong, it's you are trying to extrapolate the data on the columns and tables into a picture, which is called the forest plot, correct?
yes.
Good.
So these are the concepts. These are the which you will see in any meta-analysis results report, and summary effect. And there are two type of models which we applied: fixed effect model and random effect model. And it is decided on the heterogeneity. It is the which studies are either how much there is heterogeneity. If the heterogeneity is very large, then we go for the random effect model. If heterogeneity is very low, then we uh, go for the fixed effect model. And the difference between fixed effect model and random effect model is that for random effect model, you can generalize your results, but for fixed effect model, you cannot generalize your models. These effects will project only uh, the only that studies you have collected, and only for those variables which the researchers or doctor has uh, take into their research. So far, we always inclined to apply the random effect model because we want to generalize our results in a generic way. So we will have a confidence interval and a prediction intervals and sub group analysis and regressions, and these are the different techniques. Additional concepts in the meta-analysis: word counting, power analysis, reporting and publication bias, cumulative meta-analysis, reporting meta-analysis, and so on. And there are some assumptions. I think so. It will be very, very theoretical, and we should go for the practical steps. I think so. I have shared the slide with Dr. Edward, and he will share it with you.
Yes,
And
I'm going to share the slides now. So now that there are a lot of assumptions, it takes years to actually understand. I want to remind everyone that this is a high-level overview that Professor has volunteered to just give us, because to go into these individual concepts, the assumptions, the model, or when and why do you pick this model of meta-analysis versus the other model of a meta-analysis, it's a whole textbook. Each model has a whole textbook on its own. And if we want to spend, exactly, if we want to begin to explain why we're using fixed over random effects, or why we're using meta regression over continuous regression, we're going to spend years on talking about it. So now we've identified the data, we've extracted data. What is our next step, Professor?
Okay. So
except you want to talk briefly on these models, you could just briefly, I see that you have them. When I was going through your slides, you just want to like overview, just talk briefly on each of them, and then we move to the next step on how to then analyze the data that we got. And the question is also open for anybody in the group who wants to, it's around, who wants to ask this question. Is a round table scientific session. We don't intend to make it a monologue. We intend to also let you ask some questions, and then they will bring clarity. Go ahead, Professor Sir.
Okay. I think so. We should go for some practical steps, and after that we can come back with the slides as well, and it would be interesting to go for the practical steps. So I share with you my folder. Okay, so the first step so we have conducted this meta-analysis, and we have done all the steps, and we have all the files. The first step is basically to select your topics, and when you select your topic, you will download some papers, and because you will develop the inclusion criteria, exclusion criteria. After all these, you will have to develop your search study for different uh, uh, for different databases. You will use some Boolean operators, and you will narrow your search, and finally you will have uh, very uh, very relevant and very homogeneous papers to conduct the meta-analysis. And in this meta-analysis, we have narrowed down. After that, we have only 14 papers from 2009 to 2022. And because our year window was not too much large, and we can take the uh, uh, already published papers between in the 15 year almost. So because it's a very new topic in medical study about the stem cell therapy, and we got the 14 papers. And the second step, when you will have uh, you have successful to collect the very relevant and very homogeneous papers on for the meta-analysis. Next step is basically that is uh, uh, slow and little bit you will have to very passionate. And second step is to go through the papers. Yes, I want to share the screen to show which things we will note. So this is the Bansali's paper of 2019. And let's close. The very important step to go through the paper is to go through the abstract. And it will also, when you will download the papers, it will not only be 14, and you will, it might be possible you will have 50 papers according to your research studies. After that, you will go through the abstracts, and some paper you will see, some papers are you will have to remove. It might be possible you are according to your inclusion criteria, you have you want to study on a human model, but in the title there is no discrimination, either it's about the studies about human or the studies about mice animal model. And when you will go through the abstracts, you will see either is it an human model study or animal model study. If it is animal model study, you will remove this paper, you will exclude this paper. So uh, in our meta-analysis we are conducting about the two types of diabetes, type 1 and type 2. So it was our inclusion criteria. So according to your inclusion criteria, you will go through the papers uh, uh, initially very quickly, and you will, when you have selected final papers for the meta analysis, then you will have to go thoroughly. You will prepare a summary table in a systematic way, and you will extract the data from these numerical tables, and it depends upon which variables or which parameters you have selected. So in our this study we have selected the dose of insulin and HbA1c and we have taken the fasting blood sugar glucose, fasting blood glucose, fasting plasma glucose, and C peptide. These five are the parameters which are taken by the researcher. But in this study, you are seeing we have the data on insulin, and they what is the 1 to 10 are the patients. In the Bensali studies, the uh, 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 sample size n, which we'll put in our data, which I have already show you. N means sample size, number of patient in the study, they have taken the 10, and this is the mean and standard deviation. And we will uh, extract from this study this basically these two only these two values, which is the mean and this is the standard deviation. And there were you will see some issue with the units, because we have uh, collected units per kg per day. So this is these two value we will manage in our table, and I will show you where I have. Similarly, let's suppose I again go to another paper and I show you so yes. So this is the another article on stem cell therapy for the treatment of type 1 diabetes. Excuse me. So you have selected this paper after go through the abstract and so on, and after that you will go through the paper being uh, you will summarize this paper in a systematic way, and you will prepare the summary of the overall paper and as well as the data extraction. So these they have taken the HbA1c, C-peptide, insulin, and I was here we have the insulin, and we have the also table with respect to the follow up of 1 month, second month, and third month. And these in the bottom you are seeing these are the patients data, and they have the mean C-peptide, mean HbA1c, and mean and standard deviation of insulin. So you will extract the data in an Excel sheet with in for the parameter you have selected. So here are finding the three parameters HbA1c, C-peptide, and insulin. Mean and variance you will manage in an and sometime you you are not given the required data, numerical data in the in a tables, and you will have to extract from the figures and from this description. And sometimes the papers put the data in the supplementary materials. So you will have to go to the website where you have downloaded this paper, and you will have to find out the supplementary materials, and they may put the data, attach the table in a supplementary material, so you can download from the parent source. So it's um, requires a lot of patience, because it's a iterative process. You have to dig out your the data on your selected papers as much as you can collect. So now I show you some the Excel file where we put this data. So I show you, yes. So these we have noted adverse events per year count. We have prepared a separate file on C peptide, year wise count, and fasting blood glucose, fasting plasma glucose, HbA1c, and insulin. So, yes, I was talking about the insulin. So, I open this file, how you will put the data into the Excel file. I share this file with you. Yes. So, as we talk about Bhansali's papers, so we have collected the numerical data in this way.
I think the file is not opening.
And, are you seeing this file? I
Not
share it.
really. Just minimize it again. Yeah, we can now see it. Now we see it.
So it's Bhansali's paper, and this is the numerical data we have extracted on the parameter insulin. So, uh, and this is the data on per unit per kg per day of insulin dose of prior to the stem cells therapy, and we have noted these two numerical values. And similarly, after that, we have this data on in Jiang's paper, and which is available only for and pre-treatment and 6 month after 6 month treatment. So this here is data is available on 3 months and 6 months only, and in some papers in use paper we have the data on baseline 0 month and 9 months. We have no data on insulin in 3 months and 6 months and even in a year. And so on. In Lewis paper, you have the data in 3 months, in 6 months, in 12 months, in 24 months, and 36-month follow up. You have sufficient data. So after uh, collecting or extracting this numerical data, we will have again a very prepaid a file with respect to our software, with respect to meta analysis. So this is an initial way of the data extractions. So as you have the data in the paper, you will extract and copy or paste or just input the data into the any spreadsheet file, and and so on. And after that, we have conducted this the meta analysis with respect to the follow up. So we have two groups, which is control group and treatment group. Means we have a group of control and the patients which are uh, gone through the stem cell therapy procedure. So and we have the data on 3 months, and we now I show you the analysis file of, yes, I show you first, I show you my folder, and that be, so here you are seeing this. This is uh, difference of means meta-analysis of the difference of mean on C-peptide on 3 month follow-up. So in this, we collect the data in a single sheet which have 3 month follow-up and on control group, and in our 14 from our 14 studies, and there will no all the studies 14, there will maybe 6 or 5 studies. So uh, so I was talking about the insulin. So let's suppose after in 3 months, for with 3 month follow up, you I show you how many studies I have and my team have collected and extracted the data. So I run this meta analysis through Jamovi, which is very fantastic and open source software. So and very it's very easy. It's not require any coding. You will just put the data, and I show you how I conduct this. So yes, the movie is fun. I share it with you.
So now that we're waiting for Jamovi to open, or the software to open, a quick rundown of what, for those that are joining late, this is ING Research Academy, where a collection of doctors from all over the world come together to understand clinical research. We are doctors in various phases. Some are already in fellowship, others are trying to get into residency, and some are coming up from medical school. Today we have Professor Khalil in our midst, helping us navigate the process of how to write how to conduct a meta-analysis. We have these clinical roundtable sessions every now and then, where we get guest speakers to put us through, reduce these terms to very simplistic terminologies and help us understand it. The whole essence, the whole objective is long-term to get our patients better, make patient management more effective, understand clinical research, and improve our clinical research outputs. Professor Khalil, what is the next step? When you extract the data and then you put it, I believe Jamovi must have opened by now, the software that you're going to use to put in the data.
Okay.
You want to share your screen?
Okay, thank you. So actually, this is the table. The author Kansali, you have data on 3-month follow-up, and there was the 10 patient. As I have go with the parent paper, and after that we go to the Excel sheet, and after the Excel sheet you can put this data in any software, even in Meta Essential, in Jamovie, in Stata, in R, in some add-ins available into the Excel sheet, Meta Excel, and Meta Easy as well. So these were the table which I have shown in the slides. So this is the number of patients, the sample size, and they have found the insulin dose point 93 units per kg per day before the stem cell therapy, and after that, this is the standard deviation of the control group, and after the stem cell therapy, there is and the sample size is same, and the same patients were gone through the stem cell therapy. After 3 months, their insulin dose reduced to 0.44 with this standard deviation. And we also find this data into Hughes paper of 2013, and this is the sample size, and we have the mean of control groups and standard deviation. And after the stem cell therapy of 3 months, after 3 month follow-up, we have this is the mean and standard deviation of the insulin dose with after 3 months. So we have found the data on with 3-month follow-up in 10 studies. So now you are seeing some even in some papers, Ashwani's paper, this dose is almost similar, no reduction, and in yes. Yes, almost there is a reduction with with due to stem cell therapy, but in Ashwani's study there is no reduction even after. So our objective is to find out there is difference, and we are interested to find out the overall effect or size of effect overall. Either there is significant difference, and how much the overall difference is. So in Jamovie, we have this is the major. You will for the meta analysis there is if we have the correlation table as I have already show you. So we have only two values, R and N. R for correlation coefficients and N for the sample size. And here we will have only two tables. One will be R and other will be N. Okay. So and third column of the author and year, and we will see the overall of correlation. If there is a study where we have a data in counts in numbers, so we have given this treatment to 10 patients, and there is a positive sign in 9 patients, so it will be a dichotomous variables. If you have only a single mean study, then you will go for this one. So here in this study, we have two groups. So mean difference, you will have the mean difference. So when we click on the mean difference. So here is very obviously you will put group one sample size. So one sample size means control groups. So it's n1. I have put and group one mean. This is the mean one. Yes. Group one standard deviation. This is the standard deviation one. And group two sample size. This is the n2. And group two sample mean. This is the mean two. And group two standard deviation. This is standard deviation. And the author and years is will be put in the study label. So you are seeing Jamovi have conducted and produced these results.
Wow,
So
that's
it's a
awesome.
random fact model. Even it's a very fantastic, which is not available in multiple meta-analysis softwares because it also write
The
a descriptive
analysis,
report.
the descriptive report. Awesome, awesome. This is
Yes,
awesome.
yes, yes. Very uh, fantastic software. So you and second one is that you will just copy and paste into your Word document, and after that you will precise. I will show you that. So uh, yes, some things we have go far here. Model options and display the model fit, and which is not it's a little bit statistical. We see which model is better. We should because we have different different models, and here we have applied restricted maximum likelihood model, and some apply only maximum likelihood model, and some and you will have to you can change fixed effect or random effect. Okay, so these are the random fact, and it is restricted maximum likelihood models. You can change accordingly, which according to this AIC and BIC, archaic information criteria and Bayesian information criteria and archaic Bayesian information criteria, which value is low for the table for the model will be recommendable.
Hmm.
So it's not required in your meta-analysis. You can and after that, here is the forest plot.
So this Gmovi plots, this software, they also plot the forest plots for you automatically.
Yes, yes,
Absolutely
yes, yes.
brilliant. For those that are joining us for the first time, or those that are new in research, Professor Khalil is walking us through step by step in conducting the meta-analysis. This session was supposed to last for 1 hour, but it's going to go beyond 1 hour because this is really very detailed and very complex. If you wish to stay to the end of the discussion, fine. I'll be here sticking up with my professor, gaining knowledge as time goes. Go ahead, sir.
Thank you. So this is the forest plot, and we are seeing this here, the situation. And you will, this is the plot, and you will put the model fitting weights, and it will be, yes, this it's also showing in some way. In some studies, you also see the model weights. So you can if you can added some things, it's like some size. It will be a big forest plot, but we should be very concise and small. Is highly recommendable.
A very quick,
So
I'm sorry, Professor, a very quick disclaimer. We also want to say we don't have any affiliation with Gmovi, neither have they um, paid us for any
so
commercials for them.
it's
It's a free
it's
software.
definitely its training is another session, and we can plan it to the trainings on some meta-analysis softwares. So it's
Absolutely.
another definitely another
So I want us to
require
quickly,
another talk on.
so I want us to
So
quickly
this
jump to
is,
the next step, analyzing this data.
yes. So yes, you are seeing because our objective how either the stem cell therapy significantly i- reduce the insulin dose of the diabetic patients of both type 1 and type 2. And in our search studies, we have found 10 authors who reported the results with 3-month follow-up. And here you are seeing the overall effect. This is the point
83.
83. This is the difference, because this is the difference from control group minus treatment stem cell therapy group. So the difference is positive. It means that the in dose of insulin is reduced in overall. In some studies, you are seeing the study of
Also,
Carlson. There is increase in dose after 3 months, and there is Asfani. I have show you 19 and 19. There is was no change, so it's on 0. There is no difference. And in all other studies,
zero.
in 8 studies, it means 80% of the studies are reporting there is a decrease after 3 months in insulin due to stem cell therapy,
Mm.
and so on. This is the publication bias. It's also there are different uh, tests reported by fail and fail safe, and is basically this is the 157 counts, and it's also significant.
Yeah.
Means that uh, we to remove this fact into 0, we required 157 studies.
Mm.
So that is very high number. And the other step, which is the Bags and Mazumdar rank correlation test. This is the p value, which is the non-significant, means that there is no publication bias. Eagles regression is also reported in some papers. So it's also non-significant, means that the publication bias is not almost. Publication bias comes from different sources, because you know that in when we have the non-significant results, we uh, don't want to publish. And and the journals are also not uh, encourage the publications with non-significant results. You know that we have taken the published papers, so there is a significant results in majority. So uh, uh, and there due to this, the publication bias is inherent in the meta-analysis. So we have to calculate the publication bias. So in this study, it was non-significant. And this is the funnel plot, which is also automatically generated in this movie software in a single command. But you will have to manage the data correctly, and data extraction is very requires a very hard work. because sometimes you will have to manipulate the data with different units if there are. So in some papers we have the data on a dose of a person, of the patient, but we don't know what is the dose of the patient per day, per kg, per unit, unit per kg per day. So we have divided the dose with the weight of the respective patient, and we gain the weight of the respective patients with from, because it was in some paper it was not available, and we uh, get this weight as assessment from the BMI, from body mass index, and after that we were successful to get the patient's dose per kg per day, because we need the data in a uniform units. So that require hard work, and when you will have extract the data in a right way, and you have manage the data according to the table required in any software. So you will put in the data into the software. This is the funnel plot, and which is standard deviation is standard error. It, because you know that some studies have a very large sample size and some studies have very low sample size, and the studies which have the large sample size in statistics, it's the basic principle, the standard error will be low. So uh, uh, according to the funnel plot, we are always inclined to see all the dots in this funnel. So some studies like this and this and this are going outside this funnel due to the high standard error, and it is definitely it will be due to the low sample size. So the study which have the large sample size, the funnel standard error and standard error. Overall, we have seen the significance. The publication bias is not significant. So after this, when you have uh, yes, when you have copy these results, you will put the results into your, I show you. Yes, you will put into the Word file. So I show you, this is these are the Word files, and yes, I was talking about the insulin, and
While we're waiting for the file to open, once again, this is a collection of many doctors, who are over 200 in number, who are interested in understanding clinical research and also using clinical research to better our patient outcome. We are primarily located all over the world, from the US to Canada to Australia, South Africa, all the way to Pakistan. Today, Professor Khalil
wonderful.
will be giving us is giving us this talk on meta-analysis. We also have other talks lined up throughout the entire year, where we are focused on understanding clinical research. Professor, sir, now we've done our
Welcome.
analysis. What is the next step?
Yes. After that, you will put the results into this Excel file. So I just copy and paste into the Excel file, and I have this descriptive report, but it is not very
It's vague.
uh,
It's not very specific.
yes, it's not very specific, but it is very helpful.
Here's a guide.
So the first, yes, the first portion, if we divide this into the two portions, so from, yes, yes, this portion, it's talk about the methods of the meta-analysis. So means just like the analysis was carried out using the standardized
Method.
mean
Right.
difference SMT. This is talking about in a generic way about the
method.
methods of the meta-analysis, and this is the very specific according to the our data, because we have total k is equal to 10 studies were included in this analysis. The observed standardized mean difference ranged from this to this,
Mm.
with the majority of the estimates being positive, and the 80% I have also
Right.
talk about. There were
Studies.
only two studies which
Right.
was reporting that the insulin dose is increased, but all the studies, 8 studies were not
Right.
reporting,
Correct.
and these were the 80%, and so on. This is the uh, interpretation, you may say, in a statistical way by the movie. So here is the forest plot, and this is the publication bias, and this is the movies funnel plot. So that after that, the next step, because you are going to publish, if you put these results in with respect to the follow up of 6 months, with the 9 months, with the uh, 12 months, and you have five parameters, there will be a lot of too many papers, too many pages of the results. So we have be uh, to be very in a precise
Specific.
way, and in a
Right.
very compact way, and in a very in a compact way. So after that, I show you the final draft, uh, how we have reported these results, and we make some edits. So the next step is actually a planning, how you will represent yours. It will be different with respect to different researchers, and it will be, and you can get idea from different papers, from already published meta analysis, how to represent your results in a very concise and precise and compact way, to save the space of the uh, journals for publication. So I show you the final draft, which is yes.
While we're waiting for the final draft to load, this meeting was scheduled for 1 hour, but we way exceeded 1 hour because the professor decided to go very detailed. We're very grateful for this. For those who want to stay back to continue, this session will be recorded. For those who want to leave, that will be fine. I'll make the recording sessions available for everyone. We'll continue with the fine details step by step until we get to the end.
So this is the uh, our topic about type 1 and type 2 uh, diabetes introduction, and this is written by my team, and this is the material and method, inclusion and exclusion criteria, and this is the, yes. So you will also while you are going through the uh, uh, go through the papers, different papers, you will prepare a summary table as well in a systematic way. So it's and the second thing, you will also extract the numerical data for meta analysis. Yes, this is the, yes, I was after that I prepared. This is the work which I have done
I think your screen is
for my.
frozen. If you could minimize and also expand your screen, maybe we'll see the Microsoft document that you're referring to. Because right now we're just seeing Drive.
Are you seeing this forest plot with the corresponding 95% confidence interval?
No, not yet, but probably it's going to load. It might take a
Okay,
while. You just minimize
okay,
it and
okay.
then
Oh
expand it.
yes. Okay, okay, okay. It was showing my only folder, my folder.
Yes, you could stop sharing
So
and reshare again.
okay, okay, okay.
Good, now we can see it.
So once again, I have to go to back because I talk about this is the topic and this is the, yes, yes. You have inclusion criteria. In this, we have restricted only in two languages, Chinese and English, and clinical studies, trials involving MSCs for the treatment of diabetes. So, and similarly, you have the exclusion criteria, and these are the outcome and search studies. These are the search studies to narrow your. You will use the keywords, you will use the Boolean operators, and so on to narrow your search to extract the very specific and very homogeneous papers. And even after that, you will have to go through the abstract introduction as well. So this is the Prisma diagram. Yes, yes. I was talking about this table.
Study. Right.
When while you are going through the paper, you will manage. You will, in a systematic way, you will prepare a summary table of all the studies. And meanwhile, you will also extract the numerical data. Basically, it's not a job of a single person. It's a teamwork.
Team efforts.
And how, if you have a good team, if you have a dedicated team, then your meta-analysis will be very uh, precise, will be very efficient. It depends upon the team. If some one would like to conduct a meta-analysis and all the steps and data extraction and summary tables and search studies and all the steps by someone, it would not be, I think it would not be possible. Uh, Means there are rare some uh, people in a very low number who can conduct all these things. So because we have to report that we are not biased in this way, so we have an alternative authors to cross check
To
and to search
cross-reference.
the paper.
Absolutely.
Yes.
To minimize
So
bias.
this is a
Right.
yes, this is a teamwork, and after that, yes, no, this is the work which I have done by my team. So yes, I'm going to the forest plots with corresponding 95% confidence intervals for standard mean difference, SDM, of the insulin and funnel plots for publication bias assessment. So mm, you have seen a very large output. So I made this
Summary of it.
only, yes. And this is the funnel plot in a single, because we have the follow-ups. This is the 3-month follow-up and 6-month follow-up, 9-month follow-up, and 12-month follow-up.
Mm.
So after that, we will conclude all the summary. So I will show you next. So this is the next step for the publication. And when you have heterogeneity statistics, I have put that the test of overall effect, either the difference is significant or not. I have put the statistics, the p-value and the value of the Z statistics,
Mhm.
two
Publication
values
bias.
for the publication bias, Eagles regression and Beck's test. So after that, yes,
You do the descriptive
yes,
part.
from the results of the meta-analysis, we are talking about the insulin. So this is the insulin. Yes, according to the forest plots represented in the table 3, the overall effect size evaluated using the SDM demonstrated a substantial decrease in insulin between the administration of the MSCs
Mhm.
and the control group at the 5% level of significance in 3, 6, 9, and 12-month follow-up. And these are the results in scientific way. And so I have put all the summary of all the follow-ups in a single paragraph. So the observed
SDM.
standardized mean differences in all included studies 100% along with these uh, follow-ups were being positive,
So
which
in summary,
had indicated that a reduction in the insulin due to the MSC's transplantation.
so in
So
summary,
this
the
is the
next step
summary.
is to
Uh,
do
uh,
the
yes.
descriptive
After
analysis of
that,
the graphs.
after that you will write the conclusion and discussion. It will be completed.
Thank you, Professor. This was more than we bargained for. So more than we asked. You went to town, you delivered exceptionally.
Yes, no comment.
We would love to have you again. At this time, I want to open the floor to anyone who wants to ask any question, make a comment, make a contribution. I see so many people from Canada, from New York. Dr. Tobe is here. I could see Dr. Boba, seasoned surgeon, is also here with us. Dr. Evolve, family medicine from Canada, is here. Dr. Faithful is also here. A lot of people. Dr. Kankem Pedro, cardiologist in the making, is here with us. I could just go on and on. I know someone usually comes in with iPhone. I don't know if you can change your name from Dr. I don't want to address you as Dr. iPhone, but she was here in the last session.
Henry is Henry. Yeah.
Oh, Dr. Henry. Okay, Dr. Henry, please, you're highly welcome. And so many other people. Rita is here. Dr. Tom, internal medicine, is here from the US. A lot of them. Dr. Payne, so many people. If I skipped your name, it wasn't intentional. Dr. Choma, Dr. Jenny, Dr. Akibi, Dr. Pedro, I've mentioned Dr. Gadi, Dr. Rifayat from Nigeria. A lot of people are very interested in this topic. Questions, if you have any questions, if you don't have any questions, this is IMG Research Academy. It's actually a voluntary group when we're doing this voluntarily. It's not a paid service, full disclaimer, where we just come together as a team. We really want to understand this thing called clinical research. Why? Because we want to make our patients better. We want to see how to extrapolate this data that we're generating to translate it to better patient management. Dr. Pedro, I see your mic is on. Your mic is hot. You want to say something? Questions, comments, contributions, additions? We just
I have a couple of questions. One,
Sure.
I came late, so I'm very sorry. I just came back from, So the first question is, this Genovi, is it a free software? And or if it's not free, how can we access it? The second question I have is it just strictly for meta-analysis, or you can use it for other research, like maybe like doing like a case control cohort study or a retrospective study? Then the other thing is there like a link for transferring your data? Because most of the time the data is on Excel. Is there a way to transfer it? Is there a link up between Excel and this Genovi?
Professor Sir, you could stop the sharing so that we can return back to the general group. We have 10 minutes for this question, and then we'll wrap up for the day.
Okay.
Questions, is
So,
Genovi a free software? Can you extrapolate data?
Yes. It's a free software, and you can extract the data on your notebook even before putting into the Genovi. But it is recommendable to manage, to write in an Excel or in any spreadsheet software after that, because it will be easy to manipulate and to copy from this from Excel sheet or any other spreadsheet softwares to the Genovi. So in Genovi or in other, even you can do the meta-analysis with Meta Essentials, with other softwares I have already told, with Stata, with because Stata and R are very much complicated as compared to the Genovi. I pick the software which is very helpful for the non-statisticians. So the data extraction, you can only write, you can make the table on your notebook, and after that you will input the data into Genovi.
Thank you. Dr. Pedro, I hope that answered either partially or wholly or fully your question. More questions, comments, analysis?
That answered my question. Thank you.
Thank you so much. More questions? Dr. Chikodili, I see you. Dr. Tom, I know you're very interested in clinical research. Dr. Chama and Dr. Henry, welcome to the group. More discussions. For me, meta-analysis is one of the zeniths, one of the peak of clinical research, when you're trying to see if the result from a single study is generalizable in the general population. I know that phase 3 clinical trial also attempts to do that by increasing the sample size, but meta-analysis now takes it a further step by collecting these various results from the various studies and then measuring the effect size or the size effects of these of the result that we generated. My question is, what if you have gone through your inclusion criteria, you've generated your inclusion criteria, you've selected your papers, then when reviewing the paper, you notice that the endpoints of these studies were reported in different formats. For example, some reported you have the sample, then they reported p-value. In another study, you have the sample, and then they reported correlation, that's R, instead of p-value. I know both of them are almost interposable. And someday you have the sample size and the reported confidence interval. And then some you have the sample size and the reported, and the end value is the standard deviation. And some other studies, they did not even report either of these parameters. They just gave a descriptive analysis of their results. How do you reconcile? Can you convert and get tools that you can use to convert this endpoint data, different heterogeneous endpoint data, to be able to compare them side by side with each other? I don't know if my question makes sense.
I appreciate your question, is very nice, and but it's a very complicated question and very technical question. Because the first thing is that if you have a case report, and it means there is only one patient, and you have absolutely, obviously you have a sample size 1.
Correct.
So it will not be included into the meta-analysis, because case reports you can include into your systematic literature review, but you cannot include it for the numerical computation for the meta-analysis. The first thing. The second thing, if you have only the descriptive reports, it will also not included. And sometime you have the data in a descriptive way, and there is a table, and where you have the data into yes, 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 no, yes, then you will put you will translate this data into
Quotes.
0 and 1s. So
Correct.
it will work for the meta-analysis. The third thing is that sometimes you will have to extract, because it's very hard, you will have to collect, you are bound to collect the numerical data for meta-analysis, otherwise you are helpless. And sometimes you will have to extract the data from the graphical
Table.
from the
Representation.
graphs.
Correct.
Yes, from the pictures. Even you will have to extract, you will have to make the tables from assessing the values, numerical values from the graph. And you will have to be good understanding of the graph. And fourth thing is that if you have the data and some values are missing, like sample size is not available, but you have the confidence interval, and so there is when you will go through the Meta Essential, they have given you a table where the minimum information is blank, and you can put that minimum information, and your meta-analysis will be run. So I can show you the sheet as well. And the fifth thing is very important, is that sometimes you are why we have conducting the mean standardized mean difference. Okay, so we require the mean of control group and standard deviation of control group and sample size, and similarly into the treatment group. So in some papers you are given the median and IQR, so interquartile range, but
Right.
you have not the data on mean and variance, mean and standard deviation. So you will have to translate. So median can be treated as a mean, number one, and median from IQR, you can average out the Q1 and Q3 and Q2 uh, as an estimate of the mean,
Correct.
and you can translate the IQR into the standard deviation by dividing the IQR by 1.35, and it will be an estimate of the standard deviation. So you will have
Convert it.
to do some manipulations.
Absolutely.
So it's a very good question, and these are the hurdles and difficulties while we are going for secondary research. An important thing is that you will always try to, when you download the papers, you will also try to find out the supplementary materials. So sometimes you find the data from the supplementary material as well.
Thank you. Thank you, Professor Khalil Ahmed. Thank you to all the panel of discussants. Thank you to everyone who has joined us today. We've come to a close of the scientific session. My name is Eddie. Next month in February, we'll be having 1 or 2 sessions. Dr. Onyeka is a psychiatrist in the US who wants to talk about the anatomy of a journal, how a journal is structured. When you're looking at a journal, when you're looking at a paper, what are you looking? It's going to be an interesting time in February. Wish everyone the best. I will stop the recording at this point, and so we can return back to the ordinary session. Give me one moment.